Okay. Good morning, everyone, or good afternoon, wherever you are. Good day, good Friday. We have Emily, Richard, and Stephanie. Thank you for joining us. Really appreciate this. We're just going to wait a couple of minutes to see if we have others joining us, and then we'll get started. We have our guest is Dr. Michael Nestor this morning, and this is our first of the season, so we're thrilled that you can join us and delighted to have him as our guest. And we'll start in a couple of minutes. Okay, we'll just begin. So welcome to the Online Ethics Center's Fall 2024 webinar series, Bringing Ethics into Conversation. My name is Azita Hirsa, and I am a senior lecturer at the Rensselaer Polytechnic Institute School of Engineering, the Industrial Systems Engineering Department, where I teach courses in management of engineering and technology, ethics of modeling, ethics of engineering, and professional development, as well as enhancing, now I forget, it was a very last course I designed, enhancing critical thinking. I'm the past chair of the ethics division at the American Society of Engineering Education, ASEE, the host of the Leaders in Action conversations at Rensselaer Polytechnic Institute, and thrilled to be the webinar host of OEC since fall of 2023. The Online Ethics Center, hosted at the University of Virginia, is a digital repository of resources on teaching and learning about ethics in engineering and science. The OEC also fosters a community of faculty, students, researchers, and other professionals who come together to discuss ethics. Please check out our website at onlineethics.org. The theme of our fall webinar series this fall is engineering ethics in industry. We're continuing with the same theme as last year. I'm delighted to host our speaker today, Dr. Michael Nestor, the board director of the Government University Industry Research Roundtable at the National Academies of Science, Engineering, and Medicine. Dr. Nestor came to the academies from Johnson & Johnson, the Innovation J Labs, where he helped to drive the development of programs and initiatives to commercialize early-stage companies involved in innovative and translational biotechnology across the global health enterprise at J&J. Before J&J, Michael was director of the neural stem cell research at the Hausman Institute for Autism, where he led his own laboratory studying autism by creating brain organoids from human-induced pluripotent stem cells. His lab developed and pioneered a brain organoid model and developed novel multiplex high-throughput CRISPR and drug screening platforms, which he spun out into companies Synapsem and more recently, Autica Bio, where he now serves as Chief Scientific Officer. Michael was a venture advisor to the Momentum Fund and an early career policy ambassador for the Society of Neuroscience. He served as the AAAS Science and Policy Technology Fellow in the Office of Science, Department of Energy, where he focused on cross-cutting synthetic biology, artificial intelligence, biosecurity initiatives. Dr. Nestor recently authored the book Anticipatory Ethics and the Use of CRISPR in Humans, published by Springer Nature. This book constitutes the first anticipatory governance and policy framework for regulating the use of CRISPR to edit genes involved in human cognition. Dr. Nestor received his PhD in neuroscience from the University of Maryland School of Medicine and completed postdoctoral fellowship at the National Institute of Health, Rutgers University as an NIH IRA-CDA fellow, and at the New York Stem Cell Foundation, where he was also staff scientist. After Dr. Nestor's talk, we will take questions. To ask a question, please type it in the Q&A box at the bottom of your screen rather than the chat box, and I'll read it to him. Audience members will not be able to use their mics or cameras. Thank you so much. We're delighted to have you, Dr. Nestor. So thank you for being here, and the mic is all yours.
Thank you, and thank you for a nice introduction. Really appreciate the opportunity to speak with you and to speak with everybody in the audience listening. I'm going to share my screen now, so just bear with me for 1 second while we switch over. And you should be able to see my screen right about now.
Yes, you can see
okay,
it.
great. Thank you. Well, again, thank you for a nice introduction. It saves me the next slide, which will be an introduction slide. So that's really great. So today I'm going to be talking to you about some topics concerning CRISPR and cognitive conditions and anticipatory neuroethics. And so this really stems from a book that myself and Richard Wilson, who's in the Department of Philosophy and Religious Studies at Towson University, we worked on this together, really coming together as myself as a neuroscientist and him as a philosopher and ethicist on how we could think about a very specific case within CRISPR. So I will skip this slide because this is really a summary of what you just heard and my entire career on a slide, which is kind of interesting to do, to put your entire career on a slide. But I will really start by thinking about why we want to talk about this topic. So, CRISPR is a technology, and I'll define it in a couple minutes. But just suffice to say, for those who are not familiar with what CRISPR is, it's a gene editing technology. It's very different from the older gene editing technologies in the sense that it has the ability to do much more specific editing with what we call minimal minimal off-target effects. So we'll talk about that in a couple of minutes. But really, just want to state up front what we're going to do today, and that's really to present a sketch of what we call a practical anticipatory ethics and policy-driven framework. And we want to really build this around the use of CRISPR, but specifically in editing genes involved in cognition. So we're not talking about gene editing in general. We're not talking about cognitive enhancement or even bodily enhancement. We're really specifically talking about genes that may be involved in therapeutics development around, for example, depression or schizophrenia, or even in my area of autism. And I want to state my bias up front, and that is that I really do believe that CRISPR technology is akin to what nuclear weapons were in the 20th century and the 21st century. So these types of biotechnologies really are not well understood by the lay public. And just like we have the doomsday clock for for nuclear weapons, I think that we really need to consider that level of scrutiny on some of these biological approaches, particularly when you begin to bring in synthetic biology, which we'll touch on briefly today. I will also say my other bias. I'm a neuroscientist. I'm in therapeutic and drug development in the work that I do, not at the national academies. And I believe that CRISPR is an important technique to be used. I also think it's going to continue and it's going to expand and it's here to stay. So my argument today here is not to ban CRISPR or even to talk about how negative it is. It's to build a that brings them into the conversation, because we definitely have the potential to lose the lay public here. And once that happens, then all of the opportunities for innovation in this space will be lost. All right, so let's go to, again, just another slide on why this is really important. So, you may say, well, CRISPR technology is still kind of out there and there's not a lot being done outside of, for example, treatments for things like sickle cell disease. But you have to understand that the scientific community is moving faster than the ethics allow. And here's a great example. There's now a movement to start thinking about using CRISPR in the prenatal environment. So it wasn't already challenging to think about CRISPR in an adult, but now we're thinking about using CRISPR technology in a prenatal environment. And you can see here from the quote from the scientist in this article, the goal of the work is really to determine whether there's a window during development when it's possible to find something wrong with the fetus and treat it with an injection of DNA into cells that matter without hitting the ones that could carry those changes into future generations. So that's an important highlight I put there, because without hitting the ones that could carry those changes in the future generations, it's a nice thought, but given another technology that we'll briefly touch on called gene drive, there's going to be pressure in the market to actually to have these gene editing techniques being used across generations. So that just shows you the dimensionality of the challenge that we're talking about. Currently, right now in its state, it's very, very rudimentary and simplistic, but it will quickly blossom into a field that's going to be used. CRISPR into a technology that's going to be used across fields. So that's very much important to state up front. So what exactly is CRISPR for those who don't know? It is an acronym for what's called Cluster Regulatory Interspaced Short Palindromic Repeats, and Cas9, which again, why we call it CRISPR. It's a very highly specific, virally delivered gene editing technique. It's used to really selectively edit, so people call this molecular scissors, specific gene sequences with what we call minimal off-target effects. So why is that important? Because typically when we use traditional gene editing techniques, you may have a target gene that you're going to edit, but you oftentimes would get a gene sequence on either side of that gene that would be also edited unintentionally. And so therefore you might be trying to edit a single gene, for example, for sickle cell disease, but you may have a bunch of different side effects in terms of different mutations that you introduce or changes to the genome that you didn't expect to make when you were doing this using traditional techniques. And so therefore it was a limitation really on the technology, and really that's a completely different conversation, because you can make such specific cuts in the DNA, and you can make such specific cuts in the gene. You can essentially repair genes that are disconnected. You can also silence genes as well, so that you want to turn off. You can also turn on genes with minimal off-target effects. It doesn't mean that there are zero off-target effects. It means that the off-target effects that you may see from the editing technology are just not going to be to the level where they may have clinical significance. So this is this is one of the hallmarks of why this technology is so important. And like I just said, there are two major challenges for CRISPR therapeutics at this point, state of the art, where we are right now. And that is, still there are potential for some off-target effects. It's not completely, completely clean. And that depends on what types of genes you're editing. And it's a fairly complex, scientific question that there are a number of folks that are trying to answer now. But then also the other challenge for CRISPR is not knowing. You can imagine, if you think about human disease and you think about genetic editing, you have to know what are the functional sequences within the gene that you need to target with this CRISPR construct. And so that's difficult too, because that requires understanding the relationship between genetics and physiology, or genetics and environment. And so that's an open question that is extremely complex. However, and I don't need to say this to the audience here, everybody's heard about AI, the combination of AI technologies now with these network analysis, gene analysis, things that we do to understand the relationship between gene and environment, that is now being quickly solved. And so it's not going to be a solve in 5-year problem. It's going to be a solved in 2-year problem. So we've gone from potentially CRISPR, sort of a interesting technique, to we might be able to use it into a limited number of cases, to we're not really sure what the functional sequences of a target are, to we're going to solve this problem, and all within the span of almost about a decade. That's unprecedented for a technology like this. So that, part of this equation, which I won't touch on today, but I think is important to understand, is that we have a speed problem as well as we have, and ethics really is challenged because the speed at which ethical analysis is being done with some of these cutting edge techniques is just not fast enough, and it's really challenging. It's a really challenging situation for ethicists for sure. And then finally, the idea of addressing these issues in terms of speed, it's not just simply that it's a nice to have, but because of the complexity of how you create these networks to understand relationships between genes and function, it is required. So it's actually the convergence, and I won't touch on this today, but it's really the convergence of AI else would use it in the same way that we might use a standard therapeutic like an aspirin. So I mean, it won't be that simplistic, but that is part of the equation that we're talking about here. Again, like I said, CRISPR lacks the complexity in address diseases with now I talked about a single gene in, for example, we're just talking about sickle cell disease, which is a single gene mutation. But let's think about something like depression or schizophrenia or autism or maybe a whole host of diabetes. You can think about cancers. Oftentimes, those are what we call multi-genetic diseases. So, or multigenic diseases, meaning that they have multiple genes that are being turned on and off at any given time and interacting with environmental effects to create the disease. That's an extremely complex environment, even from a computational standpoint. So to go back to the point that I just mentioned about trying to understand the relationship between your mind without going too far, how complex that computational milieu has to look. And so that has been one of the holy grails of this technology as well, is how do we multiplex multiple genes and treat multiple genes with these really complex diseases, again, with that minimal or low off-target effect. And so that was absolutely not possible without AI. At the most, there was a targeting of 3 to 5 genes, and those genes had to be known fairly well, and there were still off target effects there. So this required the delivery of this requires the delivery of these multiplex genes require the convergence of AI and quantum computing. So I won't go too far into the quantum computing discussion today, just to say that it's this computational complexity that we have to think about. And why do we need a convergence of AI and quantum computing? Well, it's because, and I'll skip all of this text just to go to the bottom of my slide here, to say that we have to have AI to process all of this data, but then we have to have quantum computing to handle the volume of data points and the speed of computation that you need in order to create these dynamic maps of what we call genotype to phenotype environments, right? And so the last thing I'll say there is, as we are developing more and more therapeutics, I haven't even talked about synthetic biology and all of this. And synthetic biology is the ability to create new proteins or therapeutic approaches from new proteins that wouldn't normally exist in nature. That requires high levels of machine learning and AI, and that's also being done as well. Again, we can't go into fine detail because of our time constraints today, but that's a whole nother layer to this as well. So this is really the holy grail. This is the map that I think is really important to show, and that is we want to go all the way from genes all the way to behavior. We're not able to do much of that work without the role of machine learning, AI, and eventually quantum computing, and really to treat things like I mentioned, psychosis, schizophrenia, all of these really complex multigenic diseases, again, diabetes, cancers. We need these technologies. And so we are now at that point of convergence. And so I think it's high time to have this conversation. And then the last thing I'll say, because we're ethicists, many of us are ethicists here, I'm not going to engage in this conversation today, but there are some that consider this, we're converging on the idea of speciation. And that's the whole set of arguments that we could be able to really create whole new distinct populations within humans based on this. And that's a little far afield from where we want to go, but synthetic biology may play an extremely important role in that as time goes on. Drive. So everything I've been talking about, when you make a gene edit in a human being, for example, using this CRISPR technology, it stays with that individual. But let's say that you wanted to eliminate something like depression from multiple generations after that individual. It was not possible to do that. But it is now. May not at the level of complexity of a human right now, but it's being currently used in mosquito populations, for example, to take a gene of interest and to drive it through the population. That's why it's called gene drive at a much higher rate than would be predicted by Mendelian genetics. And so normally you'd have sort of a 50% inheritance rate. You could drive it up to 90% through a population, and therefore it's been used with the Zika virus to introduce genes into this mosquito population and drive them through the population so that you essentially eliminate the carriers of Zika in the mosquito population. So this has the opportunity to take it from the individual and begin to bring it in to subsequent populations. And you might want to do that in a therapeutic. Let's say we want to eliminate breast cancer, for example, for all subsequent women after the generation that we decided we were able to have a CRISPR therapeutic for. Well, then you might combine this with the gene drive, or you might introduce this in the embryo. So there's a lot of therapeutic reasons why we would use these. This isn't all sort of a doom and gloom sort of conversation. There's a lot of positive reasons why one would go after this technology. So again, just wanted to point out that gene drive is another really important aspect of this conversation we're not going to be able to get to today. So let's talk about now we're going to move into the cognitive piece of this. So the convergence of AI and quantum will solve a lot of the inherent issues with CRISPR. It's going to set the stage for CRISPR to be used well beyond its current application, and specifically to alter cognitive states. I want to set aside cognitive enhancement. I don't like having that conversation because, quite frankly, I don't think it's going to be the first use case, and I don't really know that it's a realistic use case, quite honestly. Where we really need to start is with things like treating mental conditions like this depression. So depression is the example that I'm going to use today. Let's say that you have the opportunity to treat depression using a CRISPR therapeutic. I don't think anybody would argue that you wouldn't want to try to attempt to do that. Depression is an extremely debilitating disease. It is experienced by almost all of the population, and having a therapeutic might in some sense enhance the experience of both the individual and those around them. So it seems like a normal place to sort of start in terms of trying to develop a therapeutic. And what I mean by that as well is normal, meaning not a single gene disorder. We're talking about now trying to solve them. May not even be there in a year, but we are there soon. So that's why we're having this conversation now. It's one thing to use AI-driven CRISPR screens to develop targets for diseases like cancer. So I talked about sickle cell disease. We can think about skin cancer, for example. So if I use a CRISPR screen and I develop a treatment for skin cancer and I eliminate skin cancer in the population, let's say that I'm able to even use a gene drive technology to drive that through the population. That's great, but it doesn't have the same sort of connectivity with cognitive states and our definition of personhood that, for example, something like depression would be. And why is that? Well, on one hand, obviously your skin, curing skin cancer for somebody will change their lived experience. But on another hand, we so intimately connect our cognitive states and our brain states with human experience, with personhood. And you can see in my example that that's been since the beginning of time, literally, the beginning of written time in terms of philosophy. So this isn't something I think is an arguable point. I mean, our brains are in some ways how we define personhood. So we have to treat using CRISPR techniques for genes that are involved in cognition or cognitive states as a very special and different case than we would, for example, treating skin cancer or even sickle cell disease. Some of these other diseases, I don't think we need to have a big conversation from an ethical perspective, other than from a patient's rights perspective. But this is a very different case here. And so, what is personhood? So, how will the concept of personhood be affected by CRISPR? So, we know that people have preferences, consciousness, desires, feelings, and thoughts. For example, personhood in this sense, the definition that I'm using is really this one, where the experience of pleasure and pain, being able to take moral considerations, and so on and so forth. And so you can imagine that, for example, the situation in where we're going to use CRISPR to alter a cognitive state may alter all of these things: desires, feelings, consciousness, and thoughts. A sense of time, maybe not so much, but enough of these that we can really say that CRISPR, against genes that are involved in cognition, at least, plays a really unique role. And so, from a phenomenological perspective, we can potentially redefine what I just talked about in terms of personhood as the individual, but then also all of the intersubjective lived experiences. So it's not just the lived experience of the individual, but it's their connectivity across lived experiences and from lived experiences of a whole network of individuals and stakeholders that they interact with. And that's sort of that phenomenological space that oftentimes is talked about in Husserl and others. And so if we're going to create, even if we didn't think about the gene drive piece, if we were to edit genes that are involved in depression for a single individual, you can imagine that change in that individual will have a network of changes in lived experiences across a number of stakeholders across that person's intersubjective space. And so, yes, using CRISPR to treat skin cancer can alter some aspects of personhood. Maybe that individual's daily life, the fact that they don't have cancer, for example. But using it to alter cognitive states, like I said before, is special because all in that intersubjective state will experience a change in personhood, not only the individual, but those that are related to them. And you think about that in terms of depression. Personhood is fundamental to lived experience and intersubjectivity because personhood, like I just said, extends beyond the person as a standing or status in relationship to others in a context of a social being. So again, treating an individual using this therapeutic for depression will have an effect on the context of their relationships in social settings. And so therefore, it isn't a simple individualistic decision. It's a decision that affects a whole network of other individuals. So the last thing I'll say too about this as well is that the CRISPR-mediated alteration of a present person, for example, with respect to cognitive states, even if we weren't able to use gene drive to drive it through the population, it may create a future lived experience for future persons that destabilize this common life world that we have. So this is what Husserl calls the community of monads. So this is where future persons may stand in relation to values and mutual understanding that's very different than where we stand in the current moment. So that's not just the individual and the networks of individuals in the current phenomenological space of that person receiving the treatment, but it's also the future. It's also futures persons. And so let me sort of bring this back to sort of an anticipatory ethics question. And so now that you understand all of that, it's not too much of a leap to understand that if I was an individual that took a therapeutic for depression, I would be very excited potentially to take that. But in doing so, I am participating in a situation where we may eliminate depression from the entire population and over time. And in doing that may change our whole perception of things like ethics, which derive from human phenomenological experiences. Now, how that changes, positive or negative, that's a different question. But the point is, it will change, right? Given those present day alterations that we're making. But more interestingly, I think, I wouldn't say more interesting, but I would say in parallel, another interesting aspect of this is, let me ask you a question that this is more of an anticipatory piece of this. And again, just so you understand, anticipatory ethics anticipates these innovations and how to integrate them in ethical analysis, and then also provides ethical analysis that influences technological development. So it's sort of a back and forth. And so let me ask you this ethical question that may to think about the technology with. If we were to eliminate manic depression, for example, from the population, I think many people would argue that would be a really good thing, especially for the individuals that are experiencing that. But as a lived experience of a population, um, many of our artists have manic depression or depression. So the question becomes, if we were to eliminate things, we get into this really interesting ethical space in which by doing good for the individual, we may be potentially doing harm to the lived experience of the population, for example. So, you could argue that, well, it's worth, having maybe a 20 or 30% decrease in artistic output for all of us to eliminate manic depression. That's an interesting ethical argument. You'd have to go into a stakeholder analysis to do that. But that is part of what we're talking about here. So it's not just simply the phenomenological experience that I just talked about and the technological innovation, but it's thinking about how a simple technological innovation like CRISPR has a potential societal effect that's well beyond sort of what we would think about for that individual. So again, using that idea of art and the relationship of art to depression or manic depression and how that would affect all lived experience. And then if you eliminate potentially you have a 20 or 30% decrease in artistic output across the entire population. The question is, would that change our whole perception of of the human experience? And therefore, would that then have ripple effects on things like how we develop ethical analysis? So there's multiple ways in which you can go. One of the challenges with anticipatory ethics that I'll make, or that I've heard before, and I think is important to note here, is that oftentimes people think of it as sort of creating a straw man argument or even sort of scaremongering. That's why I'm very careful to make sure that to understand that that's not the point here. The point isn't to eliminate CRISPR. It's to create sets of pieces of policy around CRISPR so that we can actually use it, but use it in a responsible way. So last thing I'll say here about anticipatory ethics. Again, it's essential to consider the lived experience by the most diverse set of stakeholders. And that's what we just talked through with that depression example that I used. And this has a functional element. So this can be done using fairly complex methods that we have exposure to now. So things like Delphi panels, focus groups, scenario workshops. These are all to get and quantify stakeholder opinion. So you can imagine this is actually a very computationally complex process if you really do it right. And so that in itself can rely on AI to make this a much more streamlined process. But the whole point is to really break that scenario that I just talked about with sort of ripple effects through a society from an individual decision, to really break that into something that we can create policy from based on an anticipatory perspective. And so we can generate future-facing models that we can subject to ethical analysis at the beginning, the end, or throughout the whole anticipatory process. Here we're kind of in the early stages, but not quite in the middle. We can use the feedback that we obtain during these analysis to be combined with what's called participatory foresight. So this is forecasting, and we can use other forecasting models to develop policy. And so this is what's typically termed as anticipatory governance. So you can see I've drawn a thread between scientific innovation, traditional ethical analysis, in phenomenology, this new anticipatory analysis, and then we're getting now to policy. So this shows how important ethics and ethical analysis can be in the tapestry of these really emerging technologies. So let me take my last couple minutes here just to talk about what our solution is and what we propose in the book. And that is what we call CRISPR Cognition Genes Database. And so what is that? So that's a database of bringing together data sources from research, preclinical data, clinical data, literature review, AI analysis, bringing that together with expert peer to create expert peer review system. And then, but most importantly to this is bringing in the lay stakeholder community, having them work together to aggregate this data from all these diverse scientific sources and perspectives to create a living database of genes that we think are important for human cognition. Because what I haven't talked about here was that we don't know all of the genes that may cause depression. And so genes may come onto that list as scientific work is done, and we may be able to pull genes off of that list as we've done research and realized they're just not really that important for editing. And so this is a living database where we really create a peer review system to bring genes on and off. And so the whole point of this is that it's a database that's constantly reviewed by experts, peers, and the lay public, and feeds back and informs additional clinical and pre-clinical data collection. And the most important part of this is that the genes in this database undergo special scrutiny, which is what we've been talking about today, and review by the stakeholders. That includes ethicists. And policy should be set to prevent CRISPR alterations of specific genes that we think might have effects that are too great on the population. And so there is a sort of a challenge here. Do we develop the therapeutic for depression, or do we say no, we're only going to go so far, because if we go past a certain point based on this database review, that we may have really strong effects on the population and future populations. So this does create a really dynamic sort of situation that requires constant vigilance and constant upkeep. Last thing I'll say is that on an international scale, this is the biggest challenge. We have to have some policy considerations that come out of all of this. I'll just read through these very quickly, but you can, I think, understand based on my conversation today why this is important. We need to develop international binding treaties enforced by international governing bodies, because we can do all this work in the U.S. and an adversary, for example, can say, I don't care about that. And I'm going to go forward with these therapeutics. And then that gets you into the whole cognitive enhancement conversation. I don't want to engage in too much today, but then we need to engage international work groups, particularly with non-science experts, clearly define unintended consequences, which the field has not done to this point, create international protocols for informed consent, de-emphasize financial incentives for using CRISPR to alter cognition. That's a huge one. And that's not happening at all. In fact, we're doing the opposite. We're encouraging companies to do this type of work. And then creating a pipeline for access for historically disadvantaged communities. Because we haven't even gotten into the conversation about who gets access to these technologies, who would get access to them first. That's a whole nother hour conversation to have. So I think I've pretty much given you a sort of 10,000-foot view of where we're at right now. I'm happy to field questions now. And then, of course, this is my contact information. I'm happy to connect with you at a later date to kind of talk in more depth. But as you can see, this is a very multi-dimensional, challenging problem. But I think ethics plays a really unique role and can play a really unique role here. The challenge is simply speed. It's the speed at which the science is being done and keeping up with it. So with that, I will stop and thank you again.
Thank you so much. This was awesome. There was so much depth, and there's so many questions I have. And for our audience, if you have any questions, please go ahead. We have about 20 minutes, so we can, Dr. Nestor can answer your questions. Just one administrative thing. At some points, you had dropped out, and so we couldn't hear everything that we were saying. And our audience are asking if it's okay to reach out to you and to be able to, as we're recording this, I think it will show up on that as well. So, and the discussion is so provocative that we want to be able to reach out to you if that's possible. And I see that you have your email there. So if that's okay, we'll go ahead and do that.
Certainly welcome it.
Yeah, so I have a couple of questions, and think of me as the 5-year-old in the room. When you talk about the lay stakeholder, community, can you define who you mean by this community?
Yeah, I mean, it's in some sense it's a fairly diffuse bucket because it involves more or less anybody who's not in the scientific community. So it would involve more or less, I guess a simple definition is, if I said the word CRISPR and you didn't know what that meant, that's probably somebody that I would like to have in the conversation, because non-scientists, I think, that's a huge challenge. And then even within that community, the broadest diversity of individuals and institutions that we can bring in, the broader that coalition is, the more accurate our sort of anticipatory analysis will be.
Okay, all right. So one of the things that I do as an educator is to talk to our undergraduates. And in fact, we have a course at RPI where we have talked about CRISPR. How do I bridge this gap between, sort of the future engineers and scientists? What part of this is necessary for me to get across to them? What are some of the things that, that are raised in your mind that, we need to make sure that the next generation's prepared? And how do I, for lack of better words, how do I water it down so that it is understood at the undergraduate level, for example?
Yeah, I mean, I think well, there's sort of two answers to that. One,
Mm,
I think that the undergraduates at this level are sophisticated enough to understand the basic concept behind gene editing and how this gene editing technique is different than previous ones. And I think just
mm,
emphasizing that part of it. So I'm using, like, I use the term molecular scissors, using terms like molecular scissors in place of, gene editing or CRISPR, that sort of thing, I think is really important to communicate. And so, and then the other part of this that I think is important to communicate is the speed, because it's not simply that it exists. It's in our lifetimes, we've seen some rapid changes in technology and rapid advances, but we have not seen the speed on the scale at the scale that we've seen, for example, with AI. So, for example, AI has been around for almost 50 years, but really it's only been in the last year that we've seen real wholesale adopting of that technology. So talking through with them that idea that it doesn't matter what the technology is, it's the speed of adoption of that technology into our daily lives without the ability to sort of respond ethically that creates the scenario where we're really challenged. And so that's the one thing. And then the other thing that I would say is, I think exposing them as much as possible to areas of work that are outside their own interests. So if you were teaching a philosophy major, for example, having them talk with scientists about this, and ask questions, I think is really important. Bringing scientists in, so getting away from the idea that we have these siloed departments and things like that, and getting much more into an interdisciplinary discussion, I think is really important, because they'll generate their own questions based on that.
yeah, yeah. Now I know you work at the juncture of government, industry, and academia. What has your experience been in trying to bring people from different disciplines, and assuming that the disciplines are different languages? What's your experience of bringing everyone to the table and being able to, come to a shared understanding? What are some of the challenges that you've experienced in what you've been doing so far?
Yeah, that's a great question. I would say that the biggest challenge that I have is one of culture. So,
Mm,
we don't have a lot of challenges, I think, in pipelining between institutions. Where our biggest challenge is in getting people to understand that institutions have very different cultures. So, for example, the word innovation is thrown around a lot,
mhm,
but my definition of innovation, if I'm sitting in an industry position, is very different than the definition of innovation if I'm sitting in academia
right.
or government. So the biggest challenge I've seen is really trying to get people to understand that when things aren't working, it's not often a process, it's a culture. And that's very difficult for people to understand, because that's not easily quantifiable, and it takes a really long time and a lot of conversation and a lot of really hard work to change. And so that's really where I've spent a lot of my time, is trying to set up systems to encourage cultural change in home institutions when you bring individuals together. And so I would say that's it. The other side of this equation, that is a challenge, is really, just specifically on the ethics and science side, is getting both of these different disciplines to be open-minded towards one another. On the scientist side, there isn't a lot of open-mindedness towards ethics. I mean, ethics is really seen as something I have to do, or I have to fill out this in my grant, or I want to make sure that we have data integrity, or those sorts of things. But it's not really thought of very fondly when we talk about these more like systemic, sort of diffuse questions that we're talking about today. And I think that's going to there's not a lack of understanding of how that potentially can lose the population when you don't do that. In fact, we have examples of how that's already been done. We can look at the COVID vaccine and the way that was handled, and the way there's such a diametrically opposed forces on that. We can look at how that was handled as a test case. On the other side, from the ethics standpoint, I just think that there's not enough of an understanding that these things are moving really quickly. And I know we all want to have really esoteric arguments about Husserl and all these really cool things. And some of these things are really important for sort of baseline knowledge in terms of creating ethical systems. But you have to move pretty quickly. And I do think ethicists have to move beyond having these what are seen by non-ethicists as esoteric arguments, and really doing much more applied work. And I think that can only help ethicists because that will get them into the conversations, which will allow them to influence the process more. And I think so. There's lots on both sides, and it just requires a lot of sort of hard work and understanding and bridge building, and and sort of a lot of self-reflection as well. Sometimes I have to think to myself, maybe I'm a little bit too much on the science side of this, or maybe I'm too much on the ethics side of this. And so it's quite a lot. And so yeah, there's no easy way to do it. You just have to do it.
Yeah. Thank you so much. And if I'm not mistaken, I believe your co-author has asked an awesome question. At RPI, we have our first quantum computer. And so we're really focused on this issue of quantum computing. And the question is, would courses such as AI across the curriculum, CRISPR across the curriculum, and quantum computing across help with undergraduate engagement? What's your answer to that?
Well, I think that quantum computing is still a little bit difficult because I don't think understanding quite yet about how we're going to use that. That might be an interesting sort of forward-looking course. I definitely think AI is, going to be something that is going to be needed. It's already, universities are playing a lot of catch up. But the challenge here is not so much catch up, it's forecasting where these AI models are going to take us and teaching and preparing our students for where they're going to be. That requires forecasting. That, I would say, requires relationships with industry, for example, with IBM and others that are developing these technologies, and asking those experts directly, where's this going to be in 3 to 5 years? Let's teach to that, and not to the questions about whether Chat GPT, for example, is going to be used in research papers and what to do about it. Yes, it's going to be used in research papers. Like that's all. We already know the answer to that. We need to start preparing students for where they're going to be, not where be reactionary. And I think that would really help with that.
That's such an awesome answer. Thank you so much. The one question that came to mind, two things. One of the things that you talked about is the culture and the speed of the culture, and the fact that we really need to take the time and have these critical thinking questions, these deep dive questions. Yet on the other hand, you're talking about the speed of all of this. How do we balance? How do we integrate? I don't know, balance is the right word. How do we integrate that time difference there?
That's a great question. That's a hard one. I mean, quite frankly, I think that we need to rethink how we are teaching students, particularly in the sense that I do think that we need to bring in more of other stakeholders into the university setting. So much more industry experts, government experts, policy experts. That's fairly difficult because obviously there's time constraints, but I do think that bringing them in alongside of the work that's already being done at the university, I think is really important. And I think that the more you can have seminars and things like that integrated with the typical coursework that you do in a semester, I think is a good place to start. But it's a hard question, quite honestly, and it's a good one. I don't have a clear answer for it.
Yeah, in terms of the playground, do you feel that the education, the academics, the academic arena is the place to have these discussions, or should it be done somewhere else? Or here, for example?
Yeah, no, it's a great question. I think the academic environment is the right one because in some sense the traditional lane of academia is this sort of bridge building, collaborative environment for learning, and I think academia is really set up well to do that. The challenge oftentimes is finding the time and getting the connections and all of that. But that's where I think places like OEC play a great role, because like, for example, today you're connecting myself that isn't in academia with folks that are. And so I think there's a multi-layered cake here. And I think organizations like OEC can play really bridge building opportunity in that layered cake. But I do think that academia is the right place to have this, especially with students as we educate the future, workforce.
Yeah, thank you. Dr. Stephanie Beard asks, as a neuroscientist and an ethicist, I find the assumptions and hubris embedded in the idea of using any technology to fix people's brains beyond removing tumors seems jaw-dropping. We need to be much more proactive at getting us all, scientists, ethicists, and members of the society, to identify and examine underlying assumptions and expectations. What are your thoughts on that?
Yeah, I mean, I understand that sentiment I do, it does resonate with me. Here's the inherent challenge of that. I agree with you. So I'll say that up front. But here's the environment where we sit in. We sit in a policy environment, an international competitiveness environment, a innovation development environment where that's being in some sense discouraged. What's being encouraged, for example, in science is how many companies can come out of your lab now. That's the new sort of
Mhm.
model that we're using. I'm sure you know that well as a neuroscientist. How many companies do you spin out of your lab? Can they develop their? I'm just talking about therapeutic development, but can they develop a therapeutic within X number of months, and then can that be rolled out to the market? I can tell you that the folks that are around CRISPR, and I know many of them well, they are trying to have an ethical conversation, a conversation about ethics. The problem is they sit in an environment that doesn't encourage that at all. And so what's being encouraged is actually the opposite, which is I wouldn't say necessarily fix people's brains, but to develop therapeutics really rapidly that then can be sold to big drug companies. It's really hard to fight against that model. And I'm not sure that's part of what I'm trying to do is not necessarily fight against the model, but educate about the dimensionality of what we're taking on here so that maybe folks like yourself, Stephanie, can go back to your home institutions and start talking about this more when, I'm sure that you've been in the conversation at the university, if you're at a university, oh, we need to have a new biopark, or we need to have more companies spun out of the university. And there's much more of a CEO approach being applied to universities now. I'm not necessarily saying that's wrong. I have a startup myself. I'm saying that we really need to examine how we're doing this at a societal level. And so that's the best I can answer. It's not a great answer, but I certainly understand.
Great. Thank you so much. I don't see any other questions, but I do have another 5-year-old question. We were talking about the stakeholders that we're trying to address, the problems of, folks with autism, manic depression, and other things. What role do they play? What position do they take at the seat at the table? What do they have to say, and how much can we explain things to them? And what I mean by that is mainly, I think, at the communication level. As a doctor, you explain things in terms of sort of the doctor language, right? The scientific language. Yet their concerns may be totally different. Their concerns may be things like, what about my family? What about this? What about that? How do we bridge that gap? How do we answer those questions for them? And what do we do? Are we there to persuade them to do one thing or another? And if not, if we want to leave it up to them, how much information, how transparent must we be in order to get the information to them?
Yeah, I mean, I think that's a great question. It's a really deep question and with a lot of layers. But let me just sort of bring the the first part of that into a model that I think works really well, which is cancer therapeutics. So, National Cancer Institute actually holds a lot of stakeholder engagements with patient groups. And in fact, during the forming of NCI, a lot of what they did was meet with patient advocates and patient groups. So I just want to mention that as a model to look at for those interested in. I would say another model to look at to answer your question is the rare disease space. And so, what is so interesting about the rare disease space is that oftentimes what you have is a lot of patient advocacy that's occurring. So, to answer your question, I think that there's a role for patient advocacy groups or patient-based advocacy groups to play this translation role and to sort of be a bridge between the more technical aspects of, for example, therapeutic development and the patients themselves. That said, the other piece that's really important is to give patients an oversized voice in this conversation. Because if you don't and you develop therapeutics without them, then again, we're kind of in an environment post-COVID where we have increasing distrust in scientific institutions. And so it goes beyond just the actual patient population themselves, but it's the idea that, we are all in some sense part of this, no matter what role we play, even if we're very far from science. And so if we're included in that conversation, we're going to be much more, I think, bought into what the results might be, or even to the research. And to make this very practical and very sort of bottom line, given what I just talked about with this innovation environment, if you don't involve patient advocacy groups and patients in this conversation about therapeutic development, you are going to lose their tax dollars, and the NIH will suffer because there'll be less and less tax money given, or at least wanting to be given to the NIH to do research. So it's really important from a bottom line, sort of based non-ethics level and economics level, to do this work. Do I think it's important from an ethics perspective? Yes, absolutely. But for those who are on the fence about it, it's a real dollars and cents question. So again, bringing them directly into
No, we got you. We lost you for a moment there.
okay,
So
sorry about that.
no, that's all right. Great. Thank you so much. We have about 5 minutes. Is there anything that you'd like to inform us of last minute? What are your last minute thoughts?
Yeah, I think so last minute thoughts are this is one aspect of what I'd like to highlight to everybody is a really interesting time, and that is we're in a time of what is called scientific convergence. And so there was just recently a popular book written about convergence, about super convergence. It's an interesting book. I don't think it meets the full mark because it's more of a popular book, but I understand the sentiment on why it was written. But it is not simply the technologies now that exist. It's not simply engineering or biology or geology. It is the convergence of these technologies and emergent properties but then also not only across disciplines, but then also the convergence of new technologies that come out. That is really where the work needs to be done. It's really on understanding what convergence is, what it's going to do, and how it's going to shape a future workforce, and how ethics can play a role in all of that. I think that's where I'm most excited about. Richard and I are working on a second book right now on convergence. We're trying to think about how anticipatory ethics can be used in that context. It's really difficult because you kind of have to forecast where things are going to go, and that's in itself difficult, especially as a scientist. I don't like to do anything without having data. So anyway, so that's where I think the future is going to be. It's on the convergence of technology and the emergence of new technologies, and then how do we redesign the university to meet these challenges, to educate students in this new space? That's what I'm most excited about right now.
Thank you so much. You certainly have my full attention. I know you and I had spoken, and I had said to you, my research originally started at the triple helix, sort of looking at the communication that happens between academia, industry, and government. And so you have my full attention. I can't wait to get my hand on the book and read it. And I know that there are those in the audience and that are in attendance that are just as excited. So if it's okay with you, at some point we'll reconvene and we'll ask you more questions if you don't mind. So thank you. Thank you so much. I also want to thank the OEC and our co-directors, Dr. Rosalind Byrne and Dr. Sergio Gwynn. And thank you for being here. Hopefully we'll get together again and we'll have this discussion, continue our discussion. Thanks again, everybody. Have a wonderful rest of the weekend and enjoy your time. Bye-bye.
Thank you. Bye-bye.